Okay, you should be good to go.
Okay. All right. Should I start now, or?
Yep.
Yep. Cool. Okay. So thank you everybody for joining. And today I want to talk about the SPAAM community and the projects we have going on. Before I start, a bit about me. I am James Yates. I'm a postdoc shared between the Max Planck Institute for Evolutionary Anthropology and the Leibniz Institute for Natural Product Research and Infectious Biology. But I specialize in ancient metagenomics with a focus on ancient oral microbiomes. However, in the last couple of years, I've been moving much more into sort of, let's say, infrastructure around ancient metagenomics. And a lot of the work is focused on the SPAAM community. And we've actually had quite a few people joining our Slack workspace from the sedaDNA community, and also our summer school and yearly meeting, which I'll mention in a minute, in the last couple of months. But so far, the sedaDNA people have been quite quiet. So I hope to introduce the projects we're working on and hopefully get more of you guys more involved. So who are we? We are a global community of people from many different institutes around the world, specializing or focusing on many different aspects of ancient metagenomics. And we're all mostly, I would say, to date, early career researchers, but not exclusively. And this includes both PhDs and postdocs and the occasional PI. And we've basically come together to really just make a very nice, open, welcoming community for the field to help work together to push the field forward in a not competitive way that many parts of ancient DNA sort of work in. And all of these images are from the various yearly workshops that we have. And we had actually one last week, in fact. So what do we do? Well, we have the yearly meetings that I mentioned, and that is meant to basically help the community to present and discuss either issues that we're having in the field and try and maybe come up with new solutions, new tools for some of these issues, but also provide a nice space where people can get suggestions and advice for their own projects, even if it's the very early stages of the project. And in that context, it is generally, so to date, has been organized by early career researchers for early career researchers. So to allow people to not have the pressure from PIs or stop PIs that may be speaking too much. And since 2020, we've been doing this remotely. So we've had three events so far. So this nice poster from Petra was done from SPAAM 3. But we're hoping we're going to revert back to our original plan, which was actually to run this as a satellite meeting, an in-person satellite meeting next to the ISBA conference. So if you're not aware of that, ISBA is a bi-yearly called the International Symposium for Biomolecular Archaeology, which has very recently been converted to a society. But that is going to now continue every 2 years, and we hope that we will have an on-off sort of in-person / hybrid or online SPAAM meeting. And so anyone can join this, and each organizing committee which cycles around decides the scope of the workshop, how it's going to be structured, who can join. So PIs often can join, but just listening. Another project we are running are the SPAAM summer schools. This was established this year, with funding from the Werner Siemens Stiftung. And this is hopefully will be a series of summer schools, not just this specific one, but this year we set it up with the Introduction to Ancient Metagenomics summer school. And this consisted of a 5-day practical workshop, or Blockpraktikum for those in Germany, aimed at master students and early stage PhD students to give them sort of a grounding in sort of an A to Z of bioinformatics workflows when working with ancient metagenomics. So what I mean by that is, this is command line, this is how you run tools all the way up through this is what your data will look like, how to pre-process it, and all the way up to things like de novo assembly, taxonomic profiling, phylogenetic trees, and so on. And all of this material has actually been made available online under a Creative Commons license. So actually, you can go to this link here, and you basically find all the slides, recordings of the lectures, and walkthroughs of all of the content that was presented. And this has funding at least to be yearly for the time being. And we're hoping that in the future, maybe there will be people running this summer school in different languages, maybe in Spanish, for example. I think there's one idea floating around, and also other summer schools for different contexts, and maybe more advanced one, maybe on field-specific things and so on. But we hope to basically provide a framework in which this can continue in a global manner for everybody in the community. And another critical thing, which why I'm happy to talk today, is that we had of the 120 people who signed up or registered to join, we could only provide 30 places, but half of these 120 people were from sedaDNA community. So there was a lot of interest from this area, but currently, so the people who were running the summer school had limited expertise, specifically in sedaDNA, because a lot of us work primarily on host-associated metagenomics. So if there's anyone interested in helping us out in the future and help train the next generation of sedaDNA researchers, please also let me know. And a third little project I wanted to mention, which also might be of relevance, is the AncientMetagenomics list. So we are trying to gather a list of all labs around the world which are working on any form of ancient metagenomics, for a variety of reasons. One is to provide a list for people maybe looking for suggestions for reviewers in the field, because we are quite a small field, but often that means a lot of people work very closely together, so finding alternative reviewers can be difficult. It can help people find collaborators for particular projects. It can help people find, let's say, reviewers not just for papers, but if they're looking for thesis advisors or external thesis advisors. And also a slightly more personal reason why we set this up for me is if you're familiar with the radiocarbon dating community, they have a really nice system where each lab gets a specific ID lab code, and which they affix to the beginning of all of their sample names. And I'm really interested in seeing if we can maybe do something similar for ancient metagenomics or ancient DNA in general, because some of the big labs, as the students move outside of their own labs, move to different groups, they've taken with them sort of very nice naming schemes for their samples. For example, in our department, we have a system where you have a 3-letter code for the site, and then a 3-letter number, 3-character number for the sample name. And other people have taken the similar concept to their labs, but now that means they're producing sample IDs which look identical to ours, and it actually has the same site code, but it corresponds to a different site. So we're now getting these sort of clashes between different labs and what sort of the codes refer to. And I suspect that this will start happening quite soon also with sort of ancient metagenomics. So I'm hoping that maybe we can set up a similar system for us as well. And anyone can basically add their lab here if they would think they should be on here, either by sending me an email or going to the website, and there's a GitHub form there. And on top of these projects, we have actually been, as a community, quite prolific, I think, given that we're only set in 2020. We've already had 2 publications coming out of SPAAM. One which was a sort of opinion hypothesis / summary from SPAAM 2 back in 2020, published in End Systems. And also, one of the things I want to talk to you in a bit more detail today, which might be of interest, is a project called AncientMetagenomeDir, which we also had published last year in Scientific Data. And so, yeah, I want to talk about 2 projects today, one being AncientMetagenomeDir, another one as well. And then basically, hopefully, have a little discussion of what would be, how to maybe get more sedaDNA people joining SPAAM, or where we can collaborate as groups and so on. But the first project I would like to talk about is AncientMetagenomeDir. And my first question is, how do you find your sequencing data? So for many people, you will probably say, I imagine, from places like NCB, the NCBI SRA, or the EBI ENA, or there's also the Japanese DBJ. I can't remember exactly what it is now. Which is generally a de facto standard for uploading raw sequencing data, particularly shotgun sequencing data, to these archives, and that's publicly accessible to everybody. But obviously, you can also find sometimes in publications other places where you can download the data. There's sometimes domain-specific databases. So in the US, there was this OGRE database, Online Ancient Genome Repository. Well, it's not been that active. Sometimes you see people uploading their sequencing data to their institution's FTP servers, sometimes Zenodo, Figshare, GitHub. So there's quite a diverse place where people upload their data. But this leads on to the question, is metagenomic data fair? And by fair, if you're not familiar with these principles, stands for findable, accessible, interoperable, and reproducible. And for most cases, I'd say ancient metagenomic data is. Paleogenomics as a whole has been actually celebrated in the past for being a poster child for public data sharing, because we generally upload all of our data to these online repositories. So this makes them accessible. Also, in genomics and genetics, we have standardized file formats, which makes it interoperable, and also that sort of helps with it being reproducible in most cases. But the findable, so the F of fair, is a bit more of a problem. And the reason why this is because the metadata around the sequencing data, so about the samples, so where it's from, how old it is, or how the data was generated, is dispersed over many, many different places. This can be sometimes directly in the main text of a publication. It can be in the SI, either in tables or the SI text. It can also be put on places like GitHub, for example. And across all of these, each author will report the information in different types of metadata. Each author will report the metadata in different formats, and each database, like the ENA or SRA, will also have its own metadata types and formats, which means how do you actually search for relevant data, comparative data, or extra data that you want to analyze across multiple places? Because the search terms that you're going to search will probably not work in every single place. And why this is an issue is ancient samples are often very rare, particularly when it comes to things like bones and skeletons, precious or also difficult to obtain, particularly I imagine in the case of like sediment DNA, if you're having to do coring, that require expensive field work and things like this. There's not something you can regrow or resample, which a lot of modern metagenomics people can use. But reusing data is still actually good for increasing sample sizes in studies, or acting as comparative data, and also generating new ideas. But you can only reuse this data if you have context. As we are probably familiar, temporal degradation means we have less and less information about our sequences. We don't know where it was from, exactly what was around at the time. We don't know what the conditions were at the time of burial, so to say. Many archaeological samples are not in situ, for example. And also, when we're doing our analyses, we really need to maximize a lot of the meta information and contextual information about the sample to account for confounding correlation factors. But if we don't have that context, it's very difficult to reuse, which then sometimes will result in people resampling what already sampled, sampling in the same area, and ultimately wasting taxpayers' money. And this is where our project AncientMetagenomeDir comes in. So this is a community-curated resource of lists of all published shotgun sequence ancient metagenomes or microbial genome level enriched samples. However, the focus is mostly on the shotgun sequence ancient metagenomes. I'll explain a bit more in a minute what I mean by that. The contents of the project is actually, or the ultimate product, so to say, is not so complicated, really. All this is a set of TSV files, so tab-separated value files, very simple tabular files, which you basically upload into Excel or R or something like that. But importantly, the way that the TSV files are generated is that they have standardized column fields corresponding to critical metadata information that is required for any ancient metagenomic sample. So this covers things like the publication information for each sample, sample name, the sample type, And like I said, this is focusing specifically on ancient DNA. So it includes a lot of metadata columns, which other contexts or databases do not have. So for example, we have age, we have the geographic location, and a couple of ways to account for heterogeneity in the way that metadata reporting is recorded about location, particularly very old samples. We have archive collection IDs, so this can include archaeological or sort of museum IDs, collection IDs, storage IDs of the samples themselves, but it can also correspond to the accession IDs in the public databases, and also URLs linking directly to the data itself, and many more depending on the context of the sort of the field of ancient metagenomics we're working on. So like I said, this is very simple. This is literally a TSV file. This is a little screenshot of the top of one of them, where we have the project name. You can see there's a standardized way of recording this, publication year, DOI, so where to find the original publications, you can cite it, the site name, latitude, longitude, and in cases where that does not exist, you also have the country ID, and there's extra columns as you go along. And the reason why we went like this rather than more complex SQL databases, web-hosted things, is that ultimately we want this as accessible as possible. So by having a TSV, anybody can download it and open it in a spreadsheet program and filter down to the period of interest or use accession numbers and things like that. You don't need to do lots of clicking in terms of finding the right web interface. You can use the web interface. Also, this is very machine readable, so you can load into programming languages and other things, which, given that we aim to be a community-based thing and have many people contributing from around the world with different computational expertise, this hopefully will make this, we design this rather to make this much more accessible. And the AncientMetagenomeDir is currently split into 3 different sections. One is host-associated metagenomes. This is typically microbiome samples. We have host-associated microbiome samples, particularly dental calculus or paleofeces, as these are the popular things in ancient DNA at the moment. We have host-associated single genomes. This is the microbial level thing that I mentioned earlier. This is for things like pathogens from the past, such as Yersinia pestis, which we see quite a lot of, like Salmonella. And finally, we've got environmental metagenome, which I imagine will be the most relevant for you guys. And this has been primarily driven by Pete, Becky, and Ibrahim. But you can already see we've gathered, as a community, loads and loads and loads of information already. So 1,000 samples for the microbiome samples, 500 for the single genome, and 300 samples for the environmental metagenome. And recently we added library metadata. And we will have almost 2,000 for these two. But you can see that comparatively, the numbers in the environmental metagenome are quite low. However, I'm pretty sure there are many more that we may be missing. But we need sort of the input from the sedaDNA community to expand this and make it really useful for you guys. And you can see from the maps that all the samples are coming from all over the world. But there is generally a like we were talking about the Jamie earlier, sort of a deficiency from sort of the global south. Although I suspect, again, there is work going on there. And as I mentioned in the previous slide, we recently expanded this to library metadata. And this is very critical, and that's something that I think is a problem for people coming from modern metagenomics into ancient metagenomics, things they don't consider. And so we include a lot of information, such as the sequencing model, because that can influence how you need to pre-process your data. We have like how deep the libraries are, because that's often common information you need to know whether your data, this public data, will fit into your data, whether download links. But we also have extra columns which are also not recorded in ENA and SRA, because this also is important for processing your data and how to analyze it. So this is stuff like library treatment. So this is damage removal, and we have another column typically called library polymerase. And the reason why these are important is, as I'm sure you're familiar, as you're all specializing on ancient metagenomics, is we have sort of 3 main problems or challenges. One is that often our true DNA, or the true original ancient DNA, is now a small sliver of the entire DNA library, because we have a lot of modern contamination that going in, either from living organisms or contaminate that living in the burial environment or the soil at the time, but also you can have contamination from the labs, from during excavation, and so on. And we also have DNA fragmentation, so our DNA is over time breaks down into smaller pieces, and the ends of these DNA molecules generally incorporate damage, so misincorporation lesions, which basically can make it more difficult for us to get precise taxonomic assignments. But at the same time, even though it makes our lives a little bit difficult, these two things, the misincorporation and fragmentation, actually is what we can use to authenticate, to show that we do actually have ancient DNA, because they have these distinct patterns. But depending on how your library has been constructed, this can be the sort of finding these characteristics in your data can sometimes become more difficult. So you're probably aware that you can run protocols such as UDG or user treatment to basically in the lab remove damage once you know that there's damage already there. But also which polymerase you select can actually also influence the amount of damage you will have. So if you use the wrong polymerase, you can basically stop the damage being retained in your library as you're doing amplification, and it means you'll result in basically having no damage signal whatsoever. And then you cannot actually separate your authentic ancient DNA from the past, whether that's 100 years ago to 10,000 years ago, from the modern contaminating DNA. And so by extending AncientMetagenomeDir to include this metadata, we hope to make ancient metagenomics much more accessible, not just to people already in the field, but people coming in from outside. But that leads me on to the next two questions. One is, how do you download your data? And also, how do you process your data? The reason why I bring up this question is many paleogenomics labs do, let's say, weird things with their sequencing. I believe sedaDNA a bit more consistent with this than other aspects of ancient metagenomics. But some labs will do things like mixing different polymerase types for all the libraries of one sample, different library types, so there's double-stranded or single-stranded, and also then sequence across different sequencing platforms as well. And all of this influences how you process your data. So sometimes in ancient DNA, we'll do one screening pass to make sure that the library did work, and then we'll do deeper sequencing, certainly in metagenomics, to get sort of the production level data we need for analysis. But when this has basically happened over this very heterogeneous setup, this requires a lot of manual work to process your data to make sure it is all actually comparable and all into one blob, which you can then analyze as saying this is one information for one sample. And this leads me on to the next little project that we've developed for the community, which is almost ready. We're doing one more hackathon in a couple of weeks, and then it will probably go live. We'll aim to publish this, called AncientMetagenomeDir Toolkit, or AMDirT, or AMDirT, depending on how you want to phrase it. And this tool is overall actually meant to be a general toolkit for anything to do with AncientMetagenomeDir, but the particular functionality I want to talk about is AMDirT filter, which allows you to load the given table from AncientMetagenomeDir into a fancy little web browser here, very much like you would in Excel, but then it allows you to search and filter in this web browser for information of interest, the samples you want. So let's say all let's say marine sediment cores from Australia. And more importantly, the tool offers you to give you scripts to allow you to download the FASTQ file. So you yourself do not have to go searching for all the URLs that you would need to download the samples you've selected, but also generate input sheets for particular ancient DNA specific pipelines, or any pipeline you would want to use it in. So for example, we have NF-core-eager, which is an ancient DNA specific pipeline for both ancient genomics and metagenomics. And a caveat, or conflict of interest, I am the developer of this pipeline. But it can be basically any pipeline that the community may want to include. And basically, the tool will generate the input sheets or YAML file or whatever for these particular pipelines for you in the format it needs to process the data, despite all this heterogeneity going on during sequencing and library construction. So this is a demo of what this looks like. You would load the tool. You can then select the particular table you're interested in, for example, in this case, the environmental one. All the different columns have filters, as you're somewhat familiar with Excel or something like that, or Google Sheets. You can then basically select by ticking the different data you want. You press the validate selection. Then the tool will basically generate in the background the download scripts. And we offer different download systems, whether that's pipeline like fetch NGS or simple curl commands. And then also the other button that was generated would provide you, as you can see here, basically an input sample sheet for the eager pipeline. So we try to minimize the amount of manual work that people have to do to retrieve their ancient metagenomic data, and then basically get it going in analysis as fast as possible by offering the download script, then the input sample sheet, which corresponds to the download script, which once you've completed, you'll have all your FASTQ files on your HPC or laptop or something like that. And then with this already prepared for you, you shouldn't hopefully have to modify this too much, and then you can actually execute the pipeline directly by giving just this as the input sheet. This is simplified, of course, but again, the purpose is to minimize this as far as possible. So to summarize this section of the talk, ancient metagenomic sequencing data and metadata is very difficult and time-consuming to find an appropriate process. AncientMetagenomeDir facilitates searching for this ancient data in the first place from the repositories by extending and standardizing and aggregating all of the metadata you need to decide which data to download. And the AMDirT tool will facilitate the retrieval and appropriate ancient DNA processing or pre-processing of the data to make sure that you can process data in a way that's consideration or has taken into consideration, the different characteristics of ancient DNA libraries. And already we have had about 30 people, as you may remember from the screenshot of the scientific data paper, including particularly Becky and Pete and Anand, who have been driving the ancient DNA section of the project. And we're really interested in, like I said, bumping up this only 300 samples up to the sort of the levels that we have for the host-associated microbiome and pathogen genomic tables for ancient DNA, for environmental DNA, eDNA, sorry. So if you're interested, please come and talk to us, because it'd be really great to have more people involved from the sediment DNA community. And particularly as that certainly seems to be the big thing at the moment. And another thing to mention, oh dear, that went wrong, but is that all of this project runs on GitHub, which is inherently collaborative. And so if you sound interested by the project, but you're a bit nervous about working GitHub, please let me know. That one of the reasons why we did this here is also to provide a sandbox or training ground for people coming into ancient metagenomics, particularly originally, at least from anthropology or archaeology, who get very little computational training, but this extends to anybody else who wants to learn how to use GitHub. This is a very nice and easy project where you can start learning how GitHub works and how you can contribute. So you can also let me know if you're interested, and if we have enough people, then we can run more of these training sessions and get you going with contributing to the project. However, this moves me on to the next project, and this is derived from the problem that actually, as much as I think AncientMetagenomeDir is great, it shouldn't really exist in the first place, because all this metadata should be with the data. And this is causing us to spin up a new project called MINAS, corresponding to minimum information about an or any ancient sequence. Because this metadata should be you should be able to find your data at the source using this metadata. It should already be linked with the metadata already in places like the ENA, NCBI, and DDBJ. And actually, to a certain extent, standardized metadata, when you upload your data, does exist, because when you do have to submit to such databases, there exists these mixes checklists, sample checklists. So you will have probably already filled these in at some point when you've been submitting your data. And these are run by the Genomic Standards Consortium, which is another sort of global group of researchers working genomics as a whole. And checklists do already exist for different contexts. And as you can see from the titles, have been published in quite high-profile publications, showing the importance to a certain extent of for such a thing. So there's ones for genomes, genomics in general. There's one for amplicon sequencing, one for sort of like a bare minimum one. We have ones for MAGs, for people doing de novo assembly, also for viruses, and also BGCs, which is a functional thing. But all of these make-sense checklists that I just described are designed for modern samples. And they've been designed in a way that some fields may not actually work for ancient DNA. So some require a mandatory collection date, which if you're working with museum samples, for example, which is maybe not so relevant for sediment DNA necessarily, but sometimes if the sample was deposited 200 years ago by some Victorian adventure or something like that, you might not have a lot of this information considered mandatory for modern samples. But also these checklists have critical information missing for ancient DNA, for example, the age of the sample, and this can be very, very important, or which particular strata or something like that it may have come from. And so what we want to do with MINAS is basically develop a community agreed upon metadata checklist that we standardize for all of paleogenomics, whether that's skeletal material, whether that's sedimentary DNA, whether that's soft tissues, usually from mummies and something like that. This checklist should basically include everything that we need to basically find or upload rich metadata that we can actually use it to make it findable by other people in the future. And so this project is in early days. So we prepared a project proposal, again, written primarily by the SPAAM community, but actually reviewed by a couple of other communities already. And we've already written a first draft of such a checklist for paleogenomics within the community, because we need this for submission to the GSC. But this is essentially what it looks like. We have, again, a sort of a title name column, a human readable name, a description of what it means, and then moving to standardization of these things, like descriptions or a schema on how each metadata field should be displayed. So like, for example, up here, we've got the sample age information. Is it like radiocarbon dated, optical luminescence imaging, and things like that? Or down here, let's say if you're dealing with skeletal material, you have the ethical information, like who authorized it, the name of the grant, and things like this. Then whether the samples are mandatory, which section it comes under, and if there's a unit that it needs to be corresponding to. So you can see it's quite standardized and very strict in the way that these things need to be formatted, but this makes it both human and more importantly, machine readable when you need to search for your data in databases. And so the next steps, which now we've prepared the project proposal and this draft sheet, is to actually get the project accepted by the GSC, which they've informally said yes already. But once they've done this formally, we can then actually start posting all of the new fields and the modification to the existing fields of the mixed checklists for discussion. And then we need to actually get the wider community involvement. We really need to get as many partners into the project as possible, and as many collaborators, to get us feedback to make sure that all of the categories that we include work for the different contexts, whether that's sediment DNA, human popgen, whatever. But also that we don't accidentally block certain metadata fields being used, and also find the missing metadata, which may be, let's say, ancient sediment DNA specific, and so on. And so we don't know yet how we want to do this, whether this be via surveys or visiting conferences or workshops, but we need to spread the word. And fortunately, we've already had some sediment DNA people involved. So Anna and Eric and Pete came to some of the earlier meetings, but the more people we get, the better. And the other reason why I want to sort of say this is important is because other than just standardizing stuff, which is normally just a lot of people jump on that, the reason why this standardization will help researchers who maybe don't contribute to the project directly, but sort of as end users, is that firstly, such a checklist will act as a template for SI tables, which means that whenever you upload your supplementary information to your publication, we hope that the field will have this template and should be in a consistent format, which means that anyone coming in will very quickly be able to look through the metadata tables as they'll be in a familiar format, which ultimately makes metadata parsing easier for readers and makes it just more understandable to know what data you have in your publication. Also, this will integrate with the ENA and SRA, because the ENA already actually interested in basically having ancient DNA specific information in their repository, because there's so much data we are producing, but currently often does not fit in. So they got lots of questions from researchers uploading the data. So they want to have something like this. So this would be great. But already the ENA and NCBI, for example, these databases provide APIs for linked data, which means that we will not just have to upload deep data here, but theoretically, people can upload their data to other databases as well and have all of these linked together. And so you can reuse such checklists with other databases. So for example, in the sedaDNA community, I believe there's this Neotoma thing, which is sort of general ecology or paleoecology, but there was potential interest for linking sequencing data. I think there's another database that Antonio Fernandez Guerra, who's one of the other main guys in the project, the Minas project, he mentioned something called Pangea. But having this Mixes checklist, or Minas in this case, will basically will allow us all to link all of these databases together to make it very easy to search across domains with other metadata as we wish. And that sort of brings me sort of to what I'd like to talk about, which is sort of the sedaDNA sort of SPAAM collaborations. So things that maybe we can offer from SPAAM, but also then you guys could bring into SPAAM or vice versa, would be things like SPAAM already provides these structured training for new sediment DNA students or any student working in ancient metagenomics via things like the SPAAM Summer School and the Git training that you get from collaborating with us in AncientMetagenomeDir. But like I said, we had a lot of people applying from sediment DNA, but just the bulk of SPAAM currently don't have the expertise on things sort of eukaryotes and sort of maybe the particularities or characteristics of your data type. SPAAM with AncientMetagenomeDir is providing a framework for ready-made databases for sequence data. So with basically a place to go and look for the data, the comparative data you may want to include to compare for whatever reason. But again, we only have a few people from sediment DNA actively in this area, so more people coming will be really fantastic because they can help propose new publications that should be added. So I'm definitely sure we're missing some, but also add the metadata and help with the reviewing of these types of publications. And then we're also looking for discipline-specific collaborators or partners to join sort of the Minas project to basically make this metadata checklist. And again, you guys could come and join and basically provide your perspectives on what we're missing. You can write your own sub-lists, as this is another part of the Minas project, to include things you think will be really important. So ultimately, it'd be really, really great to see more and more people from ancient sediment DNA and environmental DNA people coming and bringing their expertise and progress the field as a community. So share ideas, whatever problems you have, and we can sort of give outsider perspectives to some of the problems you guys might be having. And so if you're interested by any of the things I've talked about today, it'd be great to have you joining SPAAM. So like I said, a lot of people in the last few months from the sediment DNA have been joining the SPAAM Slack, but no one's really speaking. I don't really know why that is so far. So mostly people like Vilma has been around, who's in the call at the moment, but also Pete and Eric, who you should probably be familiar with. They've been speaking up, but a lot of other people have been very quiet, and it'd be great to just have you a bit more active and saying what you would like and what you need from the community as a whole. And also, if you're interested in any of the projects, you can again join the Slack channel to join in any of these. But as well as Slack, we also have a mailing list, and we're also on Twitter, so you can also follow us. So all of these are under SPAAM community, and you can always check on the website to find the links to these. And it will also be great to see, this is out of date now, this is last week, but for SPAAM 5, which we now have a committee for, it'd be great to see some of you next year, possibly at ISPA. So thank you very much, and I will take any questions.
Cool. Thanks very much for that, James. I just had one. The summer schools, are they in person or do they provide an online option as well?
Yeah, we did it hybrid, and I think we will probably continue with this. So there was quite a few people from Germany who signed up, and partly because of the funders and the people involved in the sort of setup of the summer school. So I think there was about 15 people there, but we also ran it in GatherTown at the same time. So actually everybody, even if you're in person, had to join by GatherTown, and that worked really, really well. So I think we'll continue that in the future because we do want to make it as accessible as possible. And also we're hoping that at some point we'll get to a critical mass in which we can then actually host it in different time zones as well. So there's already some interest for Spanish speakers, particularly in South America, who want to do a Spanish version of this. But yeah, the summer school I think will always be hybrid, and certainly I think that's the way that most SPAAM events, whether that's the workshops or AncientMetagenomeDir, will work, because otherwise you can't get everybody in, particularly as we're early career researchers in most cases and don't have funding for traveling themselves.
Do we have
Hi, James. Sorry, I have a question. Thank you for that talk. If we want to like collaborate with some of the SPAAM community members, who should we contact for this?
Generally go to the Slack channel and just say hi in the general, or you can contact me directly. I'm sort of the spokesperson, I'd say, at the moment. But like I said, we are very open, very friendly, and normally if you speak up on the Slack channel, everyone will jump in and we'll guide you.
Okay.
Thanks, Jay. And congrats for the Nobel Prize also. And if at all we have, it's asking, if at all we have any query regarding this ancient metagenome, and like SRA also we have, and as you said, institutional databases also we can constitute. And I just have a basic question, because you are expertise in this field. What are all the precautionary steps that we are supposed to take when we work for extracting the DNA from the sediments? Because we have to proceed for the geological analysis also. So as I went through a few of the videos, and we have to maintain strict sterile conditions. So can you give me some ideas on it with your experience?
Yeah, so I myself don't work specifically on sediment DNA, but I've worked with samples that come from excavations and things like that, so it sort of applies. It is always tricky, and you will never be able to avoid contamination, ultimately. The primary precautions that you can take is normally during during the laboratory procedures. So particularly if you're working with eukaryotic DNA, there's a lot of contamination, like strong contamination in sort of standard kits for DNA extraction. So for example, you get lots of cow DNA because of the way that they produce the reagents and things like that. So that's the main or the most comprehensive step where you can avoid contaminations in the lab by having a clean lab, wearing full body suits, all these things you would, I don't know, yeah, you would see in the Nobel Prize sort of related videos. In terms of sampling, sort of, let's say in the field, and when like you have geologists who also would need to analyze these things, the main thing would be to try and get your samples as fresh as possible before anyone else jumps in. And again, ideally where sort of, if you want to prevent human contamination, if that's an issue for you, yeah, wearing sort of masks so you don't breathe on it, wearing hair nets, maybe the bodysuits, or some form of way of stopping your cells going in there, sterilizing equipment after every sampling, ideally bring sterile tubes and things like this already. Normally people will do this either with bleach or for some of the tools, and or UVing the tubes, possibly in the lab before you bring it. But that's it. It's ultimately sort of you'll never be able to stop contamination entirely. It's more, I think, about authenticating your DNA at the other end. So separating the one contamination from the other, and it will always be a sort of a level of probability. You will never be able to get a strict yes/no answer there. So as long as you're getting things like damage patterns, short fragment lengths for your particular sequences you're interested in, you should be probably okay. It also depends on the question somewhat, but yeah, so basically be first in, use sterile equipment, And then in the lab, yeah, be super clean in the lab when you have control over that.
So whether you people will be going for any in situ preservation techniques, or do we have any publications of how to, any trial and error experimenting on the procedures that we have to undergo while handling with the ancient DNA samples or any sediment DNA samples? Do we have any publications of that sort? If you could just guide me out.
Do you mean generally all ancient DNA things, or specifically for storing in the museum?
Storage, preservation techniques, handling, whatever, the handling with sedimentary DNA, like this.
Okay, so the handling of sediment DNA, I don't know specifically, but there are quite a few publications on ancient DNA in general. So there is one by, I think the lead author is Bastien Lamas, who's actually in Adelaide. And they have A to Z public in Star, no. It's basically A to Z how to handle ancient DNA. Am I sharing my screen? Otherwise, I'll quickly Google it. Or let me check in my paper file. 1 second. And this paper is sort of the modern version of some of the old publications.
I just share a link. We published a paper with soil sediment samples recently. So yeah, you can check that one too.
Yeah, that's a good one for sediment DNA specifically. And then here we go. Star, that's the one. This is the other one. So this one I just sent is if conceptually generic, applies to anything in ancient DNA. It does focus on human DNA, but that is often the biggest contamination problem. So I would say that in metagenomics, we don't have contamination in the same way because we have slightly more ways of separating what we're looking for. Because, for example, if you're working on paleofeces, you want to look for just gut taxa. And if you've got stuff that's clean from the soil, that's fine. So I would say that the people working with human population genetics have to be even more cleaner. So if you follow their levels of contamination avoidance, that would also apply. But yeah, as Vilma says, that publication is specific to sediment DNA, and that will be helping. That's also sort of an A to Z thing from sampling all the way to analysis.
So thanks, Vilma, and thanks, James, for that. Thanks for sharing the link. And
You're welcome.
Newsletter and science magazine for this SPAAM community?
No, we don't have a sort of yearly thing yet. So the Slack channel is where most activity happens. So people just go there and ask. So we have a Slack channel called No Stupid Questions. So you can always ask literally anything you possibly want, and you won't be judged or anything like that. There is the mailing list, but it's not so active. So again, I have to say that most of our members are early career researchers who are quite into instant messaging, not this sort of more archaic forms of communication. But one project that is being set up by Shreya and Elle is a blog as well, where sort of we want to wrap up some of the discussions that maybe happen and so the advice that gets shared on the Slack channel, but sort of formalize it a bit more and have it in a bit more visible place on a blog thing. So you could also probably see that. And that will also be on the website. I think we've made that live already. Let me just double check. Yes. So that'll be here. So for example, we'll have a post upcoming episode. The first one will be on what is a cluster, because there's like 20 different definitions of a cluster, whether that's a HPC cluster, whether that's cluster factor, whether that's a cluster on the sequencing machine, which sometimes confuses people. So things like that. Another blog post that might come up is particularities about the Kraken unique taxonomic classifier, if you've heard of this. There's certain things that you have to tweak it slightly to get it to work properly with ancient, or more efficiently with ancient DNA. So someone might publish that in a blog post. So it can be anything you want, and anything you can also, I think you, Shreya and Elle said, you can also ask them. So you have this question and they will ask people to try and write a little blog post to answer it for you.
So thanks, James, for that. Thanks for guiding me.
You're welcome.
And thanks for the talk.
And hi, Barbara. Barbara has also been joining SPAAM as well.
Do we have any last questions? Cool. I'd just like to thank you again, James, for getting up super early for us.
Thanks for the invite.
It was a great talk, and it's really nice to hear about what's going on in the SPAAM community. And yeah, thanks for looking to make links with the sedaDNA-Society. I think there's a lot of great connections that can be made there. So hopefully we'll get a lot of involvement in the near future.
That'd be great. Yeah, I'll definitely send Pete and Eric to evangelize as well.
Yeah, for sure. Cool. Thanks everyone for joining. And there'll be a video of this up online shortly. All right. See ya.
Bye.